Hi everyone. Good afternoon, and we'll be starting today with the next live session 5. So, as first of all, I'll be of this course, I think biology, and here are some important links. So the first thing is the link for the uh, live session meetings, which are going on, which is this link. The second is the YouTube link, where you can find all the recordings of the live sessions. And the third thing is the Google Drive link, where all the materials for the live session, such as the PPT that you're seeing right now, will be uploaded. Yeah. Just a second, we'll get started. Yeah. So first, we'll recap lecture 24, which was the first lecture of genetics. So this week we had 3 lectures on genetics. I'll go over them one by one. So the first was genetics 1, where basically uh, we started talking about how all life on Earth is encoded for by using the DNA. So that basically means that DNA is the basic backbone of all life, and basically all instructions for life are written in DNA, irrespective of the living organism we are looking into, except for like viruses and all which use RNA. So, what are the implications of this? So, the first thing is that we are united with all the living organisms, all the living things. So, that means that all of us share the same language uh, in terms of genetics. Uh, the second thing is that much of the biology that we learn about other organisms applies to humans also. So, uh, much of whatever biology, whatever we learn about genes, whatever we learn about DNA from other model organisms such as bacteria and all. Also, bacteria, Drosophila and all, C. elegans also applies to human beings. The third thing is that DNA is a way of storing information. So basically, our genetic information, our genetic makeup is basically what defines who we are and how we are, how we came to, how we come to be basically, what all things our body can perform, what all things our body can do, and so on. So DNA is a way of storing all that information. And so looking at it that way, looking at DNA as a way of storing information, we can then say that by exchanging DNA, we can also exchange information across organisms and even across species. So yeah, so this is basically the foundation of all genetic engineering that has been done. So the simple fact that all life on Earth is encoded for by DNA allows us to do genetic engineering, allows us to basically learn from other organisms about DNA and genetics and gene information transfer and so on. Right. So here are some uh, aims. So yeah, so after this unit, the one should be able to answer these following things. So the first thing, uh, first observation is that children resemble their parents. So the question is why? And even if children resemble their parents, why are they not identical? So why are not every siblings identical to each other? Right. The second thing is that we know that genes encode certain traits, such as, for example, you'll be long, you'll be short, um, you'll have long fingers, short fingers, thick fingers, and so on. So, how do we gain the knowledge that okay, these genes encode for these traits, and so on? And the third question is, or the third thing that we should be able to answer is what are the sociological consequences of genetic engineering and genetic technology? So back even in the 1889s, 1890s. So this is from a magazine in 1889 where it's a cartoon showing basically how we have bred dogs to be very different. Like so different kinds of dogs can be bred to be very different. So the notion of breeding, the notion of heredity that we can basically selectively breed animals or selectively modify the behavior or morphology or basically the way these animals look and the things they do and so on is was very well exploited. And basically, especially in farm animals such as cows and so on, they are generally bred to have higher yields of, let's say, milk or meat and so on, right? Even goats and poultry and other animals. So the notions of heredity were very well exploited since quite a long time. Uh, but before we knew about how this heredity is carried forward. So how these traits are basically transferred from one organism to the other, to the offspring via genes and so on. We knew about this principle of inheritance. So this is credited to Gregor von Mendel, who was a friar and led a monastic life in Austria. He um, basically that allowed him to educate himself without having any financial burden, and he was trained in physics and mathematics. And using his mathematical knowledge and working on pea plants, he was able to uh, understand and basically lay the foundation for many principles of inheritance in pea plants that also apply to other plants and animals, including humans. So we'll take a look at what he found out and how he found that out. So basically, he was working with pea plants, which the scientific name is basically Pisum sativum. I'm sorry, this should be italicized, but yeah, I forgot to do that. I'll do that. So these pea plants basically have these different kinds of features that he was looking into. So, for example, the seeds can be round or wrinkled, the cotyledons of the seeds can be yellow or green. The color of the flower can be white or purple. The form of the pea pod can be like full or constricted. The color of the pea pod can also vary from yellow and green. And basically, the stem can also have characteristics such as it has a very tall plant. So the size of the plant is tall or the size of the plant is short. And also it has like flowers that are axial. So basically, this means that the flowers are present on not on the apical meristem. So basically have the stem going like this and the flowers are placed on either side of that of the growing stem or they can be terminal. So basically each branch or each meristem ends in a flower. Right. So these are the different features that he looked at in pea plants. And Mendel basically crossed these pea plants or basically made them breed with each other to get at various formulas for inheritance. So for this, he used true breeding strains. So true breeding strains are basically pea plants that consistently produce offsprings with the same traits as the parents upon selfing. So, for example, let's say you have a pea plant here, which is very tall, and let's say it is a true breeding strain, then what will happen is if you breed this pea plant with another true breeding tall pea plant, the progeny, all of the progeny will be tall. Or in another way, if you basically self this pea plant, so you basically uh, make it breed with itself, then all the progeny that it should produce will be tall. So how do you basically breed or self these pea plants? So basically, pea plants, plants in general are pollinated. So flowers are basically the reproductive structures of plants, and they contain various different male and female structures, such as this ovary, styles, and ovary, stigma, and ovules are basically the female structures. Stamen, anther, filament, and pollen are basically the male structures. So what happens is these pollens, they basically land on this mm, mm, stigma of the flower. So the pollens that uh, come from the anther or the stamen of the flower land on the stigma of the flower to basically then go inside and reproduce with the ovules that are present in the ovary of the plant, ovary of the flower. So selfing is basically when you take pollen from the same individual and put it on the um, stigma of the same individual. So may it be the same flower or different flower of the same individual, doesn't matter. So basically you take pollen from the same individual and put it on the stigma of the same individual. So that is known as selfing. And crossing is basically when you take pollen from one individual and put it on another individual, which might or might not have the same characteristics. Right. So we'll next talk about what kind of experiments Mendel did with these pea plants. So basically, what he did was he found two true breeding pea plants which had different uh, contrasting characters. So, for example, we see in this example here in the pea generation, which is the parental generation, uh, you can see that we have one pea plant, true breeding pea plant, which has violet flowers. All flowers are violet, and one pea plant which has white flowers. So all the flowers are white. And so what Mendel did was he crossed these to give rise to the F1 generation or the first filial generation. F stands for filial here. So and what he saw was that all of the progeny of these two plants, because they have contrasting characters, so the progeny is said to be hybrid progeny. So all the progeny, all the hybrid progeny have violet flowers. So they don't have any white flowers. And next, what he did was he basically celled all these F1 plants or basically bred them with each other, crossed them with each other. So F1 was crossed with F1 and then that gave rise to the F2 generation, where he saw that the white flowers re-emerge. So initially he had thought basically probably that the white flowers are basically the uh, basically people didn't know about genes back then. So the characters that carry these white flowers in these plants were basically gone, but that is not the case. So there is some way in which this white flower character of this plant is also present in this F1 generation, but it is not seen. So it's basically present but not seen. And then when you basically cross this F1 plants with other F1 plants, this white character that is present is then emerges back. So you have like around, you have plants which have violet flowers and also slightly less number of plants which have white flowers. So by doing this experiment, by repeating this experiment on multiple different plants and for uh, multiple contrasting traits. So we talked about all of these different traits before, right? The ones that are labeled here, all of these different traits, he took them one by one and he tried and did all these uh, experiments with them. So he found out that in the F1 generation or in the first filial generation, what happens is all of the offsprings almost always show one of these traits, but not both. And then in the second generation, in the F2 generation, in the F2 generation, just a second, sorry. Yeah, so in the F2 generation, what basically he sees is that the other trait, so the contrasting trait that was not present in the F1 generation, re-emerges. And he, after doing this experiment with a large number of plants, a large number of pea plants with a lot of different traits, he found out that the ratio in which these traits appeared in the F2 generation was very close to 3:1. Right. So all of these ratios, if you look at all of these numbers, if you look at, so the ratio that you get is 3:1, where 3 is the dominant character, which is seen in the F1 generation, and 1 basically corresponds to the, or the lesser number is generally corresponds to the hidden character, which is not present in F1 generation. Right. So, sorry. So, based on this, Mendel had a few ideas. He had a few um, explanation of your hypothesis. So the first thing is that he's he said that the characteristics do not blend. So if you have a violet flower and a white flower, they don't mix to give you a flower of uh, s- that has a color that is in between, right? So either you get violet flowers in a plant or you get white flowers. You don't get a mix of them, right? So the characteristics that are there do not blend. The second thing is the key connection that he made was that each parent has two copies of the character and basically transmits one of that to its offspring. So basically, what Mendel wants to say here is that this violet flowers in the true breeding strain in the parental generation are coded for by two copies of the same character. So, for example, let's say here capital V, capital V. And these white flowers, in this case, let's say small v, are coded for by these characters. And these are basically, so each of these individual transmits one of it to its offspring. So the offspring that we see here in the F1 generation will have capital V and small v. Right. And then again, like if we do that further, we see what happens. So basically, Reginald Punnett was another mathematician, I guess, who gave this way of basically Punnett square is basically the way of calculating uh, how the genes might be transferred or how these characters might be transferred from the parent to the offspring. So using the Punnett square, we'll see what happens. So basically, let's say in the parental generation, now instead of the V, we'll use P. So let's say capital P. Capital P represents violet flowers, and small p represents white flowers. Right. And so this is the parental generation where we are crossing two true breeding plants. Right. This is small p, small p. So what happens here is this violet plant will basically give capital P and capital P gametes. Right. And this white plant will basically give small p, small p. So in the F1 generation, what will happen is we'll have capital P, small p, capital P, small p, capital P, small p, and capital P, small p. So all the individuals in the F1 generation will have this uh, kind of character, which is capital P, small p. And since we know that violet is what is seen and white is what is not seen, so this violet color and this violet character, this capital P, is said to be dominant over the small p. So the dominant character is what is expressed. So all these plants in F1 generation are violet, have violet flowers. Right. Now, when we cross this F1 with another F1 plant, what happens is, so this F1 gives rise to two plants, which is small. And if we take another individual, again we have capital P, small p. Now, if we basically make this match and see, so what we'll get is capital P here, capital P, small p here, small p, capital P here, or capital P, small p. Either way doesn't work, doesn't matter. Small p, small p. So now what happens is this plant here will be violet will have violet flowers, this plant will have violet flowers, this plant will have violet flowers because capital P is dominant over small p. Whereas this one here will have white flowers because there's no capital P here. There's only small p. So what we see here is that basically in a ratio of 3 is to 1, we get these characters of violet is to white flowers. So this is what Mendel was able to show, that if you have a large number of plants, if you do this experiment, repeat this experiment with a large number of plants, because probability also plays a factor. So let's say a pea plant produces 1,000 gametes. Exactly 500 will be capital P, exactly 500 will be small p is not the case. So there'll be some probabilistic fluctuation. And that's why you have to basically take a large number of p plants so that you get a ratio that is approximately equal to this mathematically calculated value of 3 is to 1. And that is what we see here, that using all these large number of plants, he was able to get a ratio which is very close to 3 is to 1, which is not exactly 3 is to 1 in all the cases, but very close to that. So this is because of the probabilistic nature of the gamete formation, right? So Mendel, although he gave us this pattern of inheritance and how basically characters are inherited from parent to offspring, he did not know about genes, right? Because genes were not discovered back when he discovered or when he proposed his idea of inheritance. Right. We came to know about genes much later. So how did we figure out that genes are the things that basically carry these characters from uh, parent to the offspring? So this was done by someone called Thomas Morgan Hunt, who was an American scientist. So he basically used to carry out experiments with this Drosophila or fruit flies. So what he saw in the lab was that male flies are smaller and female flies are generally larger. And what he found that there were some male flies which had white eyes, and there were some other flies, male and female, which had red eyes. Right. So what he did was he basically crossed a red-eyed fly, female fly, with a white-eyed male fly, and he saw that the progeny all had red eyes, right? No matter male or female. Then what he did basically was use this female and mated it with another red-eyed male, and he found that basically he was able to get a white-eyed male. And he noted that this pattern of inheritance, so people at that time, when in Thomas Morgan's time, people knew about chromosomes. People knew that there are basically sex chromosomes, and basically females have XX chromosomes and males have XY chromosome. So basically, they have one pair of X chromosome. And this pattern of inheritance of this high color in Drosophila is very similar to the pattern of inheritance of X chromosome or sex chromosome in Drosophila. So what he proposed was that these traits, these eye color traits, are carried on the X chromosome, or basically these genes encode for these characters. Right. So that was the key connection that was made by Thomas Morgan Hunt, and yeah, basically, so explaining this further, we have two X chromosomes here on the female. Both of them code for the red eye. And since the male has only one X chromosome, which codes for white eye, when basically there's inheritance, so the males all get their X chromosomes from the female, so all of them have red eyes, and females are basically get one X chromosome from the female, one X from the male, and basically they have one uh, red and one white chromosome, and basically red eye and white eye trait, and basically because red eye is dominant, they express red eye. But when this female is mated with another male, so all the males that are produced by this female, half of the males that are produced by this female will have this X chromosome, and therefore half of the males will have white eyes, and half of the males will have red eyes. Right. So now that people knew that genes encode for traits and genes lie on chromosome, what is the inheritance mechanism from parent to offspring? So how are these genes replicated? Why are not basically all siblings equal? Right. What happens? So to look at to understand what happens, we have to take a look at this process of meiosis slightly further. So what happens in the process of meiosis is that uh, gametes are produced at the end of meiosis because there are two uh, cytokinesis events, two basically cell divisions, and only one instance of DNA duplication. So a normal human cell generally has two pairs of chromosomes. So basically two of each chromosome. So in meiosis, what happens is first DNA is copied so that we have four of each chromosome in a given cell. Then all four copies of the chromosome come together. So here we have these two chromosomes that are connected, are basically called sister chromatids. And these two chromosomes, which are basically lined up with each other in the first meiosis division, is basically called homologous chromosome. Right. So what happens is when they line up during the metaphase, they basically have a recombination where basically chromosome segments are exchanged between these homologous chromosomes. Then basically the cell divides so that these chromosomes separate out into two different cells. And because of this recombination or exchange of chromosome segments, there is slight difference between the mm, genes that are present here versus the genes that are present here compared to the initial cell from which they came. Right. So there's slight variation that is introduced into this cell because of this recombination. Then again, after the second round of cell division, we have basically four gametes. Each has one copy of each chromosome. So now we don't have pairs of chromosome. We have a single chromosome in each of the gamete cells. Right. So and each of these will be slightly different from each other because of recombination and segregation. Right. So that's one of the reasons why basically siblings don't look alike because there's some recombination going on, and all gametes that are produced are slightly varying from the other gametes that are present. Since the time of Mendel and Hunt, there have been a few important discoveries that have been made. For example, Mendel said that basically all genes uh, segregate independent of each other. So, for example, as we looked at the traits before, the violet color of the flowers should segregate independent of the yellow color of the pea pod or yellow color of the seed, right? Uh, but this person called Stutevan showed that there is some amount of linkage. So, like some traits tend to be segregated together, and some traits tend to be linked. So that's why they're segregating together, and they are found more. They are often found to be occurring more because with respect to random chance where they would be independently assorted. Then people have discovered things such as so we talked about dominant genes, for example, the violet flowers, which are dominant over the white flowers. But there are also things such as codominance, where basically uh, multiple traits can dominate at the same time. Incomplete dominance, where the dominant trait is not dominating fully. Penetrance, where basically uh, genes might or might not be expressed fully, depending on other factors in the individual, and epistasis, where the uh, character of a gene is controlled by another gene, right? The expression of a gene is controlled by another gene. Then Barbara McClintock also discovered transposons which are basically mobile genetic elements which can move between chromosomes. And then later, Watson and Crick figured out the double helix structure of the DNA. So that was it for the first lecture. Does anyone have any questions or any doubts? Feel free to unmute yourself and ask if you have any questions. Uh, just a second. Slight issue here. Uh, okay. Yeah. So there's a power cut. We'll go through the things slightly faster than expected. So in the next lecture, we again talked about genetics. So here we talk about linking genotype to phenotype. So basically, what is genotype and what is phenotype before we go into it? So genotype is basically the genetic makeup or the exact code of DNA that is present in your genes, right? And phenotype is basically what is expressed. So, what does that genotype lead to? What expression of traits does it lead to? For example, as we saw in the case of the pea plant, so the capital P, or small p is the genotype of the pea plant, and the expression that is the violet flowers or the white flowers is basically the phenotype. So, in this lecture, we talked about forward and reverse genetics. So, let's say so forward genetics is when we basically take a phenotype, and from the phenotype, we try and figure out what is the genotype of, exactly what is the gene genetic makeup. And in the case of reverse genetics, what we do is we basically take a genotype and then try to figure out what phenotype does this genotype result in. Right. So we looked at a few examples. So the first thing is the screen for embryonic lethal genes. So this is from work that was carried out by Nassim Walhead and Eric Wickshaws, where they basically took mutants of Drosophila that had different developmental defects, and then basically they tried to figure out which genes made them have these developmental defects. So this is an example of basically a forward forward genetic study where basically we take these different developmental deformations such as propel, hunchback, myops, and all, and we basically try to go from this phenotype to figure out when they basically try to go from this phenotype and basically figure out what is the genotype that is resulting in this phenotype. Right. In the second example, uh, what we basically talked about is work carried out by Robert Harvest. Uh, so basically, he worked on these worms called C. elegans. So in these C. elegans, basically you can follow this cell lineage. So each cell of the C. elegans basically can uh, divide, and basically that leads to other cells, and there's a pattern to it. So all these different cell divisions can be tracked, and you can have a lineage of cells based on which cell this cell came from and which cell the previous cell came from, and so on and so forth. So the cell lineage has around 1090 cells, but the mature nematode, which is C. elegans, has around 959 cells. So all the cells that are missing, so the 131 cells that are missing, are basically dead cells. So they basically go through a process of apoptosis or programmed cell death, where basically due to the presence of certain genes, they basically die off in the process of development. So what Harvest did was is he took a few mutant worms in which they did not have these 959 cells, but rather had more cells. And then he basically tried to see which genes were they lacking, or which genes were basically missing or deactivated in these mutated worms, leading to improper apoptosis or improper programmed cell death. So that's in that way he was able to identify the death genes that were present in this C. elegans, this nematode. So this is also an example of basically forward genetics. Mm, because we are looking at phenotype, we are looking at mutant forms, and basically going from there to identify the genes, the genotype that is giving rise to this case, right? So the interesting thing is that the human homologs of these two genes that were isolated by Horvitz have been implicated in cancer. And if you think about it, basically these cells, these genes, which basically lead to apoptosis or programmed cell death, are basically when they are mutated, then these genes cannot lead to this process of apoptosis. And basically, cells which are meant to die do not die, and basically they proliferate, leading to tumorous growths or cancer. Right. In the next example, we looked at uh, work carried out by Dr. Michael Smith, where he basically did something which is called site-directed mutagenesis. So what he did was basically he took these plasmid DNA, and basically he um, okay, yeah, he basically took these plasmids, and basically uh, what he did was he created a template DNA which contained the wild type sequence, uh, and basically modified a single nucleotide, or basically modified a single nucleotide to introduce a mutation to create a primer. So basically what he did was basically, let's say you take this template strand which has this uh, sequence of CTCGCCTTC, and he basically kept everything the same except for this uh, G, which he modified, right? And again, like he did it for different, basically both the strands, he created primers, and basically because these primers, even though they are not completely matching to the wild type genome, there's some slight mismatch, but overall they are matching with a high degree of accuracy. What happens is eventually they end up combining with this uh, template DNA and reproducing. And basically, after that, what he was able to do is he was able to get a complete plasmid which had the mutation that he wanted. So basically, he was able to mutate only a single site in this plasmid, and then he looked at the effects of this single mutation to see what happens if you introduce this mutation. So this is an example of reverse genetics, because we are going from genotype to a phenotype. We are introducing like uh, mutations in a given site and looking at what these mutations do to the organism or how this changes the phenotype of the organism. Right. Similarly, here this other example from Craig Mello and Andrea Fay, where they basically uh, came up with a method called RNA interference, which is basically um, in this method, basically they introduce a double-stranded RNA, inject a double-stranded RNA into a worm, into the C. elegans worm, and they basically see that this basically prevented the expression of certain proteins in the worm, and that led to certain phenotype or certain behaviors, such as this twitching, which is often seen in the presence of another mutation. So that's how they were able to link basically what kind of different genotype changes lead to different phenotypes. Right. So this is also an example of reverse genetics because they go from uh, genotype to phenotype. They basically change or basically stop a protein from being synthesized or modify basically the RNA behavior of the RNA and then basically see what phenotype it results in. Right. Uh, yeah. So RNA is a very important and a very what do you say? Yeah, it's a very important technique for studying different kinds of genes because you can then basically knock down different genes that are present in various organisms, such as the C. elegans, and see what is the effect of switching off this particular gene or this particular gene, and maybe a combination of these genes and so on, and basically seeing how what phenotypes are emerging from a given genotype or a given set of genes. Right. Next, we talked about the Human Genome Project, which was a landmark project in genetics. So, where basically the idea was to sequence the genome of a human being. And it was very important. It was a very important milestone event because it led to a large number of people collaborating from across the globe and with laying down different protocols for open data sharing. And also, because of the Human Genome Project, there were certain improvements, there were a lot of improvements in the sequencing technology in the years to follow. And this was the first reference genome for Homo sapiens that was assembled. And the approximate cost for this project was around $3 billion. Yeah. And basically in Buffalo, New York, they uh, got 20 volunteers from which they from whom they took blood, and using this blood they tried to sequence their genome. And this is a photo of the newspaper advertisement recruiting volunteers for the Human Genome Project. And the statistic is that 93% of the genome is sequenced from 11 of these donors, and almost 70% of the genome of the Human Genome Project is sequenced from a single donor. Right. So along the way, there have been multiple changes, multiple advances in the field of sequencing, and that has led to basically uh, accelerated growth and accelerated um, what is it? So the project completed much before than they were thinking, and also these improved sequencing techniques mean that now it is much easier and relatively much cheaper to sequence a genome. So, for example, let's say in 2001 or 2003, when this Human Genome Project was going on, when this Human Genome Project was basically running, the cost of sequencing human genome was quite high. You saw that the cost of this Human Genome Project was around $3 billion, but nowadays, in like for example, 2021 and so on, the cost of sequencing a single human genome is around $1,000, which is around 80,000 rupees, which is quite cheap. So how does this sequencing technique work? Is technology work is also something that we talked about. So the basic idea is that DNA is, as we know, it's a double helical structure. It's separated into single strands, then it is chopped up into smaller pieces and mounted on a surface, such as a bead. So, for example, we have a bead here, and we have multiple single-stranded DNA strands that are present on this bead. Each of these strands, each of these fragments serves as a template, and the template then guides the synthesis of a complementary strand. Right. So complementary strands are basically created from these templates. Right. With complementary bases that are subtitled one at a time. Addition of each of these bays releases a specific kind of light, or a specific color of light. So we have four bases ATGC, and addition of each different one releases a light that is of a different color. And then we have a camera or a detector that records the light, color of the light that is released, and the order in which the colors are detected reveals a sequence of the original strand that was present. Yeah. So there is two kinds of basic sequencing techniques. So the first is Sanger sequencing. So this was devised by someone called Frederick Sanger, who won a Nobel Prize for this. So basically, what he did, he basically took genomic DNA, and basically for a particular region of interest, a particular region where you want to sample the genome, you basically cut it out and you basically amplify that region using something called a polymerase chain reaction, or a PCR, which we'll learn about in the later lectures. So once you amplify this region of interest, what you then do is you basically give a lot of these fluorescently labeled nucleotides, which DDNTPs. So these are basically nucleotides that uh, stop the process of elongation of these complementary strands. So what happens is, if you have a template strand and you have like different kinds of nucleotides that are coming up, and if you have a fluorescently labeled nucleotide that joins, then basically the elongation of this strand is stopped here, and so the strand is basically uh, this much long. And again, for another example, we might have strands that are shorter or strands that are longer, right, depending on when the fluorescently labeled DDNTPs bind to the complementary strand, right? And then basically the fragments are then separated based on their size, and the DNA sequence is read in the order in which the fluorescent signals are detected. So basically, if the smallest strand, let's say, gives out this, 1 second. Huh. So if the smallest strand gives out the signal of, let's say, yellow, that means there's a T there. And if, say, the next one gives out the signal of blue, that means there's a C there. Then if you get a red, that means there's a, and so on. And based on, so this is based on the length of the strand. So this is the smallest, and this is the largest here, going from the smallest to the largest. So we read a sequence based on the size of the DNA strand that is sequenced, where you have the color, right? Fluorescent color that has emerged. So this Sanger sequencing is very appropriate for short length DNA strands. And for longer length DNA strands, what we have is something similar but slightly more evolved, which is called second generation sequencing, also known as um, second. Sorry. Also known as next generation sequencing, next or second generation sequencing. So what happens in this case is that, as we talked before, in the slide before, basically the genomic DNA is chopped up into different smaller segments. Then there are some adapters that are added to the ends of the DNA. These adapters basically help it bind to solid surfaces such as these beads, and where they're basically amplified. And in this case, fluorescently labeled nucleotides are incorporated, while highly sensitive cameras record the sequence of lights. So these basically have four different fluorescently labeled nucleotides, which basically give rise to four different colors. And basically, when each of them are basically getting uh, added to the complementary sequence, what happens is they emit a different color of light, and there's a camera that basically records these sequence of lights, and therefore we have a sequence of DNA. So then, because we have chopped up the DNA into very small fragments, what we have is now many different small fragments that are uh, the sequence of which is now known. And basically, what happens is if you have a known genome, known reference genome, such as a known bacterial reference genome, what you can do is you can basically align the sequence reads that you get. So each of these smaller sequence, uh, mm, the for each of these smaller sequence, you get all of these uh, basically sequence. The nucleotide sequence for each of these smaller reads. You can basically align it to the reference genome, or you can basically try and map it based on the different sequence reads that you have to give rise to a reference genome. Right. So this is next generation sequencing, which is appropriate for sequencing larger strands of DNA, whereas Sanger sequencing is appropriate for smaller strands of DNA or RNA. Now the next thing that people want is like in 2021, we have we can easily genome sequence the whole genome for $1,000, but the future in the future, can we do it for much cheaper? For example, let's say $100. Right. So this would amount to around 8,000 rupees for a single human genome. So the major issue, the major limiting factor here is not the sequencing technology, but the number of sequences. So, for example, let's say you're going for a clinical study, to sequence a single genome, it's much more expensive than to sequence, let's say, 100 genomes or 1,000 genomes, because the cost tends to scale down as you increase the number of genomes that are to be sequenced. So, basically, if in the future we have a pipeline, or basically maybe due to development of more cheaper sequencing uh, techniques, maybe cheaper sequencing equipment, we might be able to achieve this goal. Right. And right now, what is possible, which is incredibly interesting, is that there's something called a single cell RNA-seq, where you can basically sequence the RNA from a single cell, and you can basically see what part of the genome is expressed in that given cell. So this is a map of single cell RNA-seq from different cells of the human body. And here you can see in the zoomed out part, for example, you can see that cardiac muscle and skeletal muscle are close together because they express similar kinds of RNA in their cells, which makes sense because both of them are muscle cells compared to any other cells that are slightly further away, for example, the nervous system cells, or basically other cancer cell lines, or other hepatocytes, which are cells found in the liver, right? So the single cell RNA-seq, what it basically helps us do is it basically helps us uh, get at what proteins are exactly being encoded in a given cell at a given point of time, and how we can basically tap into that to uh, basically see how different cells function, how different cells cluster together based on the functions that they carry out, or they separate from each other based on the functions that they carry out, even though all the cells have the same genomic material, that is DNA. The RNA is what differentiates them, right, based on their function. So does anyone have any questions based on that? Sorry for the bad quality of the video. There's a power cut, so I'm not sure how long I'll be able to record. If we stop somewhere in between, because my laptop battery is also getting finished. If we stop somewhere in between, I'll record the rest of the lecture and upload it on YouTube when the power comes back on. Okay. So if no one has any doubts, we'll move on to the next lecture, which was again on genetics. So this is the final lecture on genetics. So here we started again by talking about the improved cost and accessibility of sequencing the human genome. So as of 2022, hundreds of thousands of people have had their genome sequenced for research or medical applications. Meanwhile, genome scale data such as SNPs or SNPs, which we'll talk about in a short bit, have been collected for tens of millions of people. So what does this mean for humans is that we are now able to improve. um, We are basically now able to sequence our genomes much more easily, much more at a much more reduced cost, and that leads to a lot of things. So the first thing is that we talked about that in the Human Genome Project, blood was collected from various uh, volunteers. So nowadays that is not the case. You don't need to have your blood taken for your genome to be sequenced. Nowadays, what you can do is you can basically even spit in a tube, and based on your spit, they can sequence your genome, and this can lead to the development of the figuring out of basically making of your family trees, getting to know your ancestry, and also perhaps getting to know any genetic predispositions you might have towards certain diseases, such as certain types of cancers or certain types of other genetic diseases. Then genome sequencing also gives rise to basically prenatal genetic screening, which can provide us with early detection of chromosomal abnormalities that might be present in a given child and probably help us mitigate those. Then genomic sequence helps in basically patient specific cancer treatment, right? Then in the clinic, basically techniques such as CRISPR, which is basically a method of um, as we saw in the insight directed mutagenesis, what uh, we were able to do is we were basically able to mutate a single site in the plasmid DNA. So using CRISPR, what we can do is we can mutate a specific segment of the genome and basically maybe remove mutations or put in or change the sequence based on what we want it to be. So genome editing using CRISPR-Cas is about to change medicine, where we can basically be able to get rid of genetic mutations that cause different kinds of genetic diseases, right? Mm, That were earlier not we, that we were earlier not able to solve. Right. Now, it's just an update. The power came back on, so we'll continue as usual. How much data is present in the able to think we can see that a single human cell carries the equivalent of about 1.5 GB of computer data in its genome, in its DNA, and the number of genes that are present in around 20,000 genes. But the thing is that it's not something that scales according to size. For example, E. coli generally has the bacterium which has genome size of 5 MB and it has around 4,000 genes, whereas Arabidopsis, which is a flowering plant, has a very meager genome size of 135 megabytes, but carries more genes than us humans, which is around 27,000 genes, right? And if you were to look at the largest genomes, we would find that the spruce tree and salamander, axolotl to be specific, have the largest genome sizes, which is around 20 GB and 32 GB, and also some of the largest number of genes, around 28,000 to 23,000. Right. So since this data can be so much volumetric, can be so big, how do you analyze this data? So by the use of bioinformatics, which is basically which lies on the interface of all these different kinds of sciences, such as biology, computer science, engineering, mathematics, and statistics. So one example of what bioinformatics do is when we look at something called SNPs or single nucleotide polymorphisms. So basically, SNPs are how we differ from each other as human beings. So, for example, let's say you have this sequence taken from two individuals. Might have basically the same genomic sequence, but there might be a single nucleotide which is slightly different. For example, here in this time, the two alleles that might be present in two different human beings might be A and G. So in one human, we have a A, and another human, we have a G. Or if you're looking at the reverse strand, it might be a T and a C. Right. So since everyone has two pairs of a gene, or two chromosomes, one we get from our mother, one we get from our father. So everyone in a population will have basically a pair of these. So let's say, for example, here is a sample population showing the different SNPs that they have. So for example, let's say this individual has a GG, this has a AG, AG, and so on. So now, if for this population we are able to, we were trying to see what is the frequency uh, of the A allele, we'll see that this is 8 by 20, which is around 40%. So we can denote it again as P. And let's say we again wanted to calculate what is the frequency of G, it will be 12 by 20, which is around 60%. So this is again called as P Q. So here we see that P plus Q equals 1, right? So because you can either have A or have a G, because we are looking at only the forward stand here, right? There's no T and C involved here. So you can either have an A or have a G. So the proportion of A that you have will always be, and the proportion of G that you have will always add up to Q1, right? So this is an example of where we had to basically find the frequency of a SNP in a small population, right? Now imagine you have to find the number of SNPs across a whole genome of human beings, across a whole large population of human beings. So now that is the kind of thing that bioinformatics basically deal with. And to do this, what they do is basically they have different kinds of models that they basically use. So, for example, here is a example of something called a Hardy-Weinberg model. Hardy-Weinberg model. So, let's say here again we are talking about the same SNP. Let's say we have a pool of parents which might have, let's say, AAGG, AGG, and so on. So, let's say of these for the kids, half of them will get an A and half of them will get a G from their mom, and half of them will get a and half of them will get a G from their dad on an average. So basically, since earlier we basically denoted the frequency of A as P, so the proportion of child which have AA will be P into P, which is basically P square. Then for AG, it will be P into Q, which is basically PQ. For AG, again, it will be P into Q, which is PQ. For GG, it will be Q into Q, which is Q squared, right? So the probability of these would be of the kids having the genotype AA would be P square, AG would be 2PQ because it occurs twice and both of them have PQ. And for GG would be Q square, right? So this is assuming that the probability of A is P and the probability of G is Q in the initial population of parents, right? So what Hardy-Weinberg principle tells us is that P square equals to Q. So this is basically one, and basically these are the proportions that you will get in your child gene, in the genotype of your children, right? So this is basically a model. So what biointerpretations do is that basically take the data that is out there, so the genomic data, then they try to fit in models such as this Hardy-Weinberg model and see if there is some deviation from this model. And if there's a deviation, they try to understand why there is a deviation and basically what can be done so that um, so what is basically changing from this model, or is this model not correct? Do we need to follow some other model and so on? So they try to basically match the observation that they see from genomic data with the model and try to see what is different and why it is different. Great. Does anyone have any questions or doubts till now? Okay, great. So uh, So genes or genomes might have many kinds of variations in them. So these variations might be small scale, large scale, or intermediate. So some small scale variations that we might have are things such as SNPs or single nucleotide polymorphism, where basically a single nucleotide is different between two individuals. We might have indels or insertion deletions, where a few base pairs are either inserted or deleted between two individuals or between two. Yeah. And we also might have short tandem repeats or STRs, which is basically uh, base pair, a set of small base pairs that is repeated a small number of times. So generally in the range of 1 to 100 base pairs. Right. We might have intermediate to large variations such as VNTRs or variable number tandem repeats, where basically we have a variable number of these tandem repeats in different individuals. Or we might have very large scale changes, which might be uh, greater than 100 base pairs to around 1 mega base pairs at 10^6 bases. Right. So here we might have basically deletion of a few genes. For example, this D and E might be different genes that are deleted from certain individuals. We might have duplication where basically we see that the gene here CD is duplicated. So we have now CD, CD. So we have it twice. There might be inversion where basically the CDE, the sequence is inversed. So now we have CDE like this, and the rest of it is similar. Then there may be complex structural variation that may be present in these genes. Right. So there are these many different kinds of variations that may be found from one human to the other. And basically looking at these variations, looking at these SNPs and all these genetic variations also helps us determine evolutionarily what is close and what is far away. So the as we move forward, now, the sequence of humans, the genomic sequence of humans is similar to chimpanzees or bonobos, more similar to chimpanzees or bonobos compared to, let's say, an orangutan. And that's how we know that humans are closely related to chimpanzees or bonobos than to an orangutan. And the gorilla comes somewhere in between. And that's how basically we make these evolutionary trees, these tree of life, based on the genomic sequences of different organisms. Right. So early humans basically started out in Africa and moved into various different places across the globe. And we also had different species of humans that were present in all these different areas. So, for example, one prominent species of humans which the sister species of Homo sapiens is Homo neanderthalis or Neanderthals, which were basically generally found in the Eurasian region. And we also had Denisovans, which were found in the Strangler area of Australia. Right. So there's a lot of diversity, even humans, even now. So we see that in terms of genetics, there's a lot more diversity in Africa compared to the rest of the world. And this might be because from Africa, where humans originated, only a part of the genomic pool traveled outside, whereas most of it has remained inside. And therefore, there's a lot more diversity inside Africa. And the Thousand Genome Project basically tries to address that and tries to sequence basically different genomes for our modern humans and tries to see if there's any difference, is there any similarity between them. And it has found by sampling 186 Africans and 184 Eurasians that no single locus of the human genome has 100% difference. So that means that we are more similar to each other, even though we might look very different, we might feel very different, but we are similar to each other on a genomic level. Right. Based on this genetic data, we can also tell when certain changes in the genes occurred and when basically different lineages diverged from each other. For example, we know that around 5 to 7 million years ago, we diverged from gorillas, whereas somewhere around 800,000 years ago, we diverged from our closest cousins, human Homo cousins, which are Neanderthals and Denisovans, and around 600,000 years ago is when Neanderthals and Denisovans diverged from each other. Right. So uh, we basically also, based on the sequence, we can tell basically how the human lineage has evolved over time. And we see that the Homo erectus eventually gave rise to Homo heidelbergensis, which gave rise to Neanderthals and Denisovans, and also as northern humans. Right. So now that we know that we can sequence our DNA and we can basically get at all this information about evolution, all this information about where do different kinds of people come from, where do we fit in the larger context of the world, and so on. So, how does our knowledge of our history inform us? So, it's very important for us to answer these ethical questions to basically try to uh, think before posing any questions, try to um, basically address these questions in better ways so as to we will not reinforce any of the biases that we might have towards specific cultures or specific people from specific regions of the world. So what is the application of all this genetics, of all this sequencing? So some examples are we can screen for diseases in newborn children. So often genetic screening can help us determine if the child is more likely to is likely to have any of these genetic diseases such as phenylketonuria, congenital hypothyroidism, cystic fibrosis, and so on. And uh, again, the ethical questions that arise here are there any negative consequences about of knowing about the baby's genetic status before it gives birth? So, is that more likely to lead to a termination of pregnancy, or is there do people uh, not like some kinds of basically babies based on the genetic status that they have? Right? So these are the negative things, negative connotations that might be associated with this. And so as a society, as basically people who are basically funding the research. So most of the research in genetics and most of science is funded by public people who are paying their taxes. So they also should be involved in all these kinds of discussions and all these kinds of ethical questions. And probably we should have like forum where everyone can raise their voice and voice their concerns about all these things before research in a certain direction proceeds. Or basically before, basically have a say in what kind of questions are asked or what kind of research is done. Another interesting application that we talked about, that ma'am talked about, is something called mitochondrial DNA sequencing. So to understand this, so mitochondria basically has a slightly different DNA than eukaryotic cells that are present. So nowadays we are able to even sequence the mitochondrial DNA, and uh, so there's this thing. So basically, a sperm only has a nucleus and it merges with a ovum from a female to basically give rise to a zygote. Right. So the cytoplasm of the zygote comes only from the maternal cell, and the nucleus comes from both the mother and the father. Right. So therefore, there's only maternal inheritance of the cytoplasm. And that means that the mitochondrial DNA that is present in a child is also inherited only from the mother. So if the mother has some mitochondrial diseases, what it means is that the child is 100% guaranteed to have that disease because the mitochondria will be inherited from its mother. So therefore, to solve these kinds of things, what people have done using techniques of in vitro fertilization, IVF, what people have done is basically now it's possible to have a three-parent baby where you basically take a donor, female egg, and you basically have a mother, female egg. You basically take the nucleus from here and the cytoplasm from here and combine them. Right, in vitro in a test tube or something, and basically then that is fertilized by a sperm, give rise to a zygote. Which basically has the donor's cytoplasm, the mother's nucleus, and the father's nucleus. Okay. So this helps prevent uh, mitochondrial disease inheritance, and this is how basically three-parent babies are uh, born using IVF. But it looks like in the future, due to enhances in techniques such as CRISPR, mitochondrial gene editing might as well be done soon. And basically, we might not have the need to have these kinds of three-parent babies where mothers have diseased mitochondria. So as I talked before, using CRISPR, we can edit the mitochondrial gene. We can also edit the gene of the baby using CRISPR. So if the DNA can be edited, what prevents designer babies from being born? So designer baby basically means that let's say you want your child to be tall. So by using CRISPR or something, you can basically modify the gene that codes for the height of child so that you get a baby that is tall, right? Uh, or for example, you want a baby that has some specific color of eye, for example, blue eye or something. You can use CRISPR to basically edit the gene of the baby and get a baby which is that specific color of eye. So if the DNA can be edited, what prevents these kinds of designer babies? And the other question that one might ask is, are designer babies bad? So some of these questions are basically, so these things are not yet widely adopted, widely used by everyone. But once we have this CRISPR technology that is widely available for everyone, which helps in like preventing a lot of diseases, helps in preventing, helps in like genetic screening and modifying the genetic makeup of diseased children to help them lead like healthy lives. Uh, what eventually people might end up doing is having these designer babies. So as a society, we need to first ask this question: are designer babies bad? And then what should they be prevented? Should they be like? Is it okay to have designer babies or is it not? And so on. Like, so these are the kinds of ethical questions that come from these kinds of research, these kinds of advances in genetics, and basically as a society, we may have to deal with these kinds of questions much sooner than we thought before. Okay, so that's the end of that lecture. Does anyone have any questions? Great. So we move on to the next lecture where we talked about gene mutations and genetic disorders. So the learning objective of this lecture was to compare point mutations and large chromosomal abnormalities and describe the differences between different kinds of mutations, such as silent versus nonsense mutations, and identify a few commonly known genetic disorders and their causes. So what is a mutation? First of all, so a mutation is basically a heritable change in the DNA sequence of an organism. So any kind of change in the DNA sequence that can be inherited by the progeny is known as mutation. Such an organism with altered DNA sequence is called a mutant. And so the mutant may be easily detectable because sometimes mutations lead to morphological changes that manifest in the phenotype. So, for example, someone might be tall, someone might be short due to a mutation, right, compared to the wild type organism. So it becomes very easy to identify a mutant. Um, Another example is like looking at black versus albino coat color in animals or change in petal color in plants. Right. So what causes mutation? So there might be spontaneous mutations, which basically are changes in the DNA sequence as a result of errors during DNA replication, transcription, or other such processes. Or there might be or mutations might be induced due to exposure to certain chemical substances such as ethyl methionate sulfonate and ethylene bromide. So these are reagents that people generally handle in labs, and if they're basically spilled on you or basically intake, uh, you intake into your body, these might lead to induced mutations. Or one more example is radiation. For example, UV rays, X-rays, and gamma rays generally lead to mutations. So that's why generally it's said that UV rays present in the sun lead to skin cancer, right, due to induced mutations in the skin. Right. So all cells possess DNA repair enzymes that try to minimize the number of mutations because most mutations are generally um, harmful to the human, harmful to the body if mm, it's not anything, right? So therefore, most all cells possess DNA repair enzymes that try to minimize the number of mutations that are present in us. Right? So what are the effects of mutation? So if we try to remember the central dogma, what happens is DNA through the process of transcription is transcribed into the mRNA or the messenger RNA, which basically then translates into a protein which may perform a function in the cell. So a gene often encodes for information to form a protein, and proteins are basically biomolecules that carry out a whole range of different cellular functions. And a mutant DNA sequence may result in a misfolded or an unfunctional protein. And this can result in alteration of phenotype and changes in the behavior, cellular behavior, and basically lead to uh, different kinds of uh, deteriorating phenotypes. Right. So there are two broad types of gene mutation. So the first one is point mutation. So where basically there's mutation in only one or a few adjacent base pairs. And then there are chromosomal aberrations where basically several genes in a chromosome are affected. And it could also involve the insertion or deletion of an entire chromosome, as we will see further. Right. So there are different types of point mutations. So the first is base substitutions. Mm, and basically where one base is substituted, one nucleotide base, which is the ATGC, adenine thymine guanine cytosine, is replaced by another. So that is something called a base substitution. And indel is basically when a lot of these bases are inserted or deleted at a given position. Right. So talking about base substitution, as we talked before, base substitution is where one base is replaced by another base. It is called a transition. So these bases are of different types. So adenine and guanine, because of structural similarities, they're called purines because they have this kind of structure. They have two ring structure. And cytosine and thymine are basically called pyrimidines because they have this single uh, ring structure. Right. So transition is basically when a purine is replaced by another purine. So for example, an adenine replaced by a guanine or a guanine replaced by an adenine is a transition. Cytosine replaced by a thymine or a thymine replaced by a cytosine is a transition. Transversion is when a purine is replaced by a pyrimidine or vice versa. So when C is replaced by G or A or T is replaced by A or G, it's called a transversion. Right. Now, based on the chemical nature of the base substitution, it can be categorized into three different types. So these types are only applicable for protein coding genes. So these are chemical nature of the protein, based on the chemical nature of changes that produce in the protein coding genes. So base substitutions can either be synonymous, also called as silent, non-synonymous, also called as missense mutations or nonsense mutations. So silent mutations are basically mutations which have no effect on the protein sequence or the protein structure. So these mutations can often remain undetected because there is no phenotypic change, and that's why they're called silent mutations. So for example, here you see that in the second case, this GTA is replaced by GTT, which also codes for a valine. So there's actually no change of phenotype because the valine is again not changed, even if there's a change from A to T, right? So this is called as a silent mutation. Missense mutation is when it results in an amino acid substitution. So this can affect the protein structure and or the protein function. And here's an example. So basically, let's say in the sequence we have uh, CCC here, which codes for this amino acid called proline. And basically, if one of the C, the first C is replaced by an A, now the amino acid that is present here will change to threonine. So what happens is the sequence of amino acid, serine, valine, proline, tyrosine changes to serine, valine, threonine, tyrosine. So the protein structure and the function of the protein can also get affected. So that's why this is called a missense mutation. Right. And it results in an amino acid substitution or change. We can also have nonsense mutations. So what happens is basically one amino acid is substituted by a stop codon. So basically the C, if it changes into a G, we now have a TAG, which is a stop codon. So here we have a protein which has serine, valine, proline, and tyrosine. But here we have a protein which has serine, valine, and proline. It does not have the tyrosine or anything that follows. So now we have what is it called a truncated protein. Therefore, this is called a nonsense mutation. Indels are basically insertion or deletion, where one or a few bases are either added or removed from the sequence. Indels often lead to something called frame shift mutations, which we'll look at in the next slide. So the frame shift mutation leads to um, so basically if we see here, so this is what a frame shift mutation is. So there's the initial unmutated sequence where you have AUG, CCU, UAU, UAC, and so on, which codes for different kinds of proteins, different kinds of amino acids. Now let's say you have an insertion of a G here in this position. So between this C, C, and G, you insert a G here, right? In this case. So now what happens is the reading frame, because we are always reading the codons are basically triplets of uh, these nucleotide bases, right? So once it starts with AUG methionine, the next one we read as CGC instead of CCG. So this produces another protein instead of this proline. So this arginine, right? And we see that the black here represents that all the proteins have been changed from here. Right. So what frame shift mutation leads to is basically a shifting of the frame in which the codons are being read, and that basically results in an amino acid sequence that is altered from the initial sequence, and this can lead to either a truncated or a non-functional protein, and which is very deleterious and often causes harm to the cell. So how do these genetic mutations or gene mutations lead to genetic disorders? So genetic disorders are basically diseases caused by a change in the DNA sequence. Genetic disorders can be caused by mutation in a single gene or in multiple genes. And environmental factors also affect the expression of genes, and they can result from large-scale changes in those chromosome structures, such as changes in the number or structure of entire chromosomes, which are also known as chromosomal aberrations. Right. Now we'll look at some genetic disorders. For example, here we look at sickle cell anemia. So this is a form of inherited blood disorder where basically the blood cells, the red blood cells, turn into a sickle kind of cell. Mm, So the shape of the cell changes and they are basically more sticky and curved, and this prevents a smooth flow in the blood vessels. So this is caused by a missense mutation, which is a base substitution single base substitution in the beta hemoglobin gene. Actually, beta hemoglobin gene. So where you basically the normal gene has a GAG which codes for glutamic acid, which is replaced by a GUG. So this A is changed into a U, and this basically leads to a valine. Sorry, instead of this glutamic acid, which leads to this disease. Similarly, we have genetic epilepsy, which is a neurological disorder characterized by seizures. Genetic epilepsy is inherited from parents to offsprings, and again, a missense mutation in the voltage weighted sodium ion channel gene, SCN1A, causes these seizures. So, a normal individual has a AAA, which codes for a lysine, which is replaced by a ACA in the mutated individual, which codes for a threonine. Right. We'll talk about all of this in more detail in the next section. Huntington's disease is an autosomal dominant disorder where basically there's a mutation in the gene called Huntington, which leads to protein aggregates in the neurons, thus killing the neurons. It leads to brain damage. So the mutation causes excess repeats of the CAG codon in the Huntington gene. So here we come across an interesting term called autosomal dominant disorder. So what this means is that, so autosomal basically means that the gene is inherited from the autosome, not from the sex chromosome. So we have basically in humans 44 plus 6 chromosomes, right? 46 chromosomes total. So autosomal disorders are inherited from these 44. What this means is that uh, if your um, parent has two normal copies of the gene and another parent has a normal and a mutated copy, and basically, you get a normal copy from this parent and a mutated copy from this parent. What it means is you will have the disease because the mutated copy is dominant. Right. Again, like if you look at it, this will produce gametes of only N, this will produce gametes of N and M. So there's a 50% chance that you will get NF and you will have a mutated. Uh, okay. gamete, a mutated chromosome that basically leads to the disease. There are many more genetic disorders involved in humans. Some are autosomal, such as Huntington disease, achondroplasia. Others are X-linked, such as hemophilia, Duchenne muscular dystrophy. So these are inherited in a X-linked manner, as we saw in the Drosophila before with Thomas and Morgan. And then there are polygenic disorders such as hypertension and diabetes, which are controlled by multiple genes. So polygenic means multiple genes. Polygenic disorders are basically controlled by multiple genes that are found. Then the next big thing is chromosomal aberrations. So basically, chromosomal aberrations are changes in the chromosome, which could involve addition or deletion of an entire chromosome and affect several genes. This can be due to a deletion. So you might have a basically chromosome. There might be some deletion where we basically have certain segments. It's like here, which are deleted. Right. There can be duplication where you basically have a chromosome that, for example, this region here is, let's say, duplicated. Right. And you might have also translocation. The translocation is basically like inversion, where basically this part of your genome comes here and this part of your chromosome comes here. There's a translocation. Right. Down syndrome is basically characterized by a gain of an extra chromosome. So now you have 45 plus XY instead of 44 plus XY. So this is also called as trisomy of the 21st chromosome because now you have three chromosomes or a triplet of chromosomes, triplet of the 21st chromosome, which leads to Down syndrome. So Down syndrome leads to distinct facial features, intellectual disability, and developmental delays in individuals. Early intervention approaches can help manage the syndrome. There's also another chromosomal aberration that leads to a disease called Turner syndrome, which is a congenital condition affecting women. So one X chromosome is missing here. So we have 44 plus XO, and basically instead of XX, you have XO because there's no chromosome there. And this leads to growth and developmental defects in affected individuals. Okay. Does anyone have any questions or doubts? Great. So we'll move on to the final lecture where we talk about studying human genetic disorders using transgenic animals. So what is a transgenic organism? A transgenic organism is basically an organism which carries foreign DNA sequences from another organism. The foreign DNA sequences are introduced into the cell using genetic engineering. Transgenic animals can also be animals that contain modified genomic sequences of the same individual. So if you take out the genomic sequence of an individual, modify it, and then reinsert it, that's also called a transgenic organism. There are different transgenic model organisms that we have, such as bacteria, yeast, C. elegans, Drosophila, mosquito rodents, such as mouse and rats, different cell lines, and also organoids, which are like simple organ kind of structures that are used to study different kind of cellular functions and changes by making these transgenic changes in them. So there are multiple research areas that involve transgenic animal models, such as Drosophila models, which look at circadian clock circuitry, which also look at cellular functions and development. We can also look at disease morphology and cure, for example, mouse models of Alzheimer's, Parkinson's, and epilepsy and cancer. And there can also be preclinical drug or vaccine testing on these transgenic animals. So animals which might have genes that are introduced from humans, we might give them drugs or certain vaccines and all and see what is the effect of that vaccine or gene on that transgenic gene, vaccine or drug on that transgenic gene that is implanted from humans, right? So uh, we'll talk more about the use of transgenic animal models to study human genetic disorders. So, what is the broad approach? So, the broad approach is that we identify a disease causing gene in the human. Then we basically create a transgenic model, made be a Drosophila, made be a mouse or a rat, might be a cell line, where we basically change the genome to reflect the changes in the disease causing gene that is present in the human. And basically, then we evaluate the disease pathology in these transgenic models. So we can look at, as we talked before, the cellular mechanisms of disease development, how in the cellular level these diseases manifest. We can study the functioning of affected cells and tissues. We can also do drug screening and novel therapeutic things to see what works better for treating this disease. Right. So, as a specific case study, Professor talked about the use of transgenic animals to study human epilepsy. So this is a very distilled form of the lecture uh, because it's a very specific research talk that was given. So I would suggest that you guys go and look at the actual lecture to know in more detail what is happening and what is going on. Here, basically, I'll give you a brief of what they figured out, what they found out, and what they did. So I would strongly recommend you go back and listen to the lecture to know more. So epilepsy is a seizure disorder. So it affects 65 million people worldwide, and about 12 lakh people in India have active epilepsy. There's a lack of awareness and a lack of healthcare services because of that, and there's absolutely no cure. So many patients are refractory, but like time and time again, it might come. So 40% of epilepsy cases are symptomatic. So that means they're caused by things such as uh, brain trauma, injury, infection, or a stroke. And 60% of cases are idiopathic. So that means they're caused due to genetic mutations, and there's no other symptoms that occur. There's just seizures that. As we talked about before briefly, so sodium ion channel gene is a hotspot for epilepsy mutations. So basically, in neurons, we have something called action potential, which is basically the way these neurons fire or give information, pass on information to other neurons. So this action potential is basically dependent on these sodium ion channels that are present on the membranes of the neurons. So when these sodium ion channels activate and deactivate, these lead to different uh, changes in the neuron, basically electric potential of the neuronal cells, which leads to these action potentials. Right. So therefore, any mutation in these sodium ion channel genes is a hotspot for epilepsy because this leads to irregular patterns of neuronal firing, which basically leads to seizures. Right. So in humans, I think we have these four domains of uh, so this gene SCN1A basically codes for these four domains of sodium ion channels, where you basically have these different four repeating structures, and basically the marks here show which new missense mutations or truncating mutations are present in this gene, and greater than 1300 mutations in this single gene alone that cause epilepsy in humans. Right. That's why it's a very important hotspot for epilepsy mutations, and it's a very um, important, like it's very important to study this gene. Right. So why do these mutations in SCN1A gene cause seizures? Basically, seizures are caused due to increased neuronal firing, which can either be due to reduction in the inhibitory neuronal activity. So there might the neurons which basically regulate the neuronal activity. So there might be there are some inhibitory neurons, there are some excitatory neurons. So the inhibitory neurons generally uh, keep the excitatory neurons in check, right? So inhibitory neurons, excitatory neurons. So these basically have to be balanced in some sort of way so that your brain functions normally. So in case of these mutations in the SCN1 A gene, what might happen is basically there's a reduction in this inhibitory neuronal activity, or there's a increase in the excitatory neuronal activity, which leads to seizures, or there might be a combination of both. Right. Epilepsy is a spectrum disorder. So what this means is that uh, as the mutation goes from missense to large number of deletions or truncation, the mutation severity increases and the seizure phenotype also increases. So the small missense mutations lead to mild symptoms such as febrile seizures, which are seizures that are accompanied by high body temperatures, or seizures that are present during fever. So episodic seizures during fever. Then in moderate cases, we might have Jeff's plus, which is something called as genetic epilepsy with febrile seizure plus. And in severe cases, when there's a truncation of these sodium gated channels, we might have something called Dravet syndrome, which is a very severe form of epilepsy. Right. So what we focus on, what we focused on in the class is Jeff's plus disorder, where basically, which is genetic epilepsy with febrile seizure plus. This is caused by missense mutations and it is autosomal dominant inheritance. So basically, very high chance that this is passed on to your offspring. And homozygous patients are not reported in humans. There's an early childhood onset, and seizures can persist into adulthood. Seizures are often associated with fever. That's why they're known as febrile seizures. And patients with Jeff's plus show pronounced clinical variation in seizure phenotypes. Some of them might have much higher uh, frequency. Some of them might have lower frequency of having a seizure. Right? Febrile seizures are basically hallmarks of Jeff's plus epilepsy. Uh, Basically, they are fever-associated seizures. Greater than 70% patients have febrile seizures, and they can last up to 6 to 7 years of age. Febrile seizure plus, they can persist beyond 7 years of age and well into adulthood. So, in this example, we talked about, Ma'am talked about research that was carried out by her uh, by using transgenic mice to study epilepsy. So they basically briefly I'll give an overview. So they basically identify the mutation in human SCN1A gene that changes a missense mutation that changes lysine to threonine. Then they use CRISPR-Cas9 to basically mutate the same gene in mice, mutating the lysine to threonine to produce transgenic mice. Then they basically uh, so try to look at what happens in the mice. So here, basically, on the left-hand side, we see how these transgenic mice were produced. We see that basically these uh, pregnant mice were uh, fertilized oocytes were injected with this CRISPR-Cas9, and basically they were bred with wild-type mm, females, and basically then they saw that after a lot of crossing and so on, they found a lot of back crossing. They found that there were two variants of this mouse that exhibited the given mutation. And basically, they took this B6NJ variant and carried out their study further, and they show that basically this is on the right hand side. We see this is the wild type genome, and this basically on the bottom we see the mutated genome. right? The sequence. So the mice groups that were studied were of three types. So the first is a control, which has a pair of normal genes, which are not mutated, a heterozygous mutant, which has one mutant gene and one normal gene, and a homozygous mutant, which has a pair of mutant genes. Homozygous mutant mice have a very short lifespan. So basically, within around a month of um, giving birth, most of them died. Right, all of them died actually if they were homozygous. Heterozygous mice and the wild type mice were surviving for a much longer time, which might also explain why we don't have homozygous patients in humans because it's a deadly disease. Right. So the for a mutant mouse model, a transgenic mouse model, to be a good model, we need to see that the behavior that is expected to be seen in humans is also seen in these mouse models. So the question here is, do these mutant mouse models have seizures like in Jeff's plus patients? So if you go to this link here, which I have provided here, you can see the videos that were shown in the class that show basically that these mouse have spontaneous and heat-induced seizures, right? Uh, when later they basically tried to figure out what was happening in the neurons of these mice, they figured out that firing in the inhibitory neurons is selectively impaired. So this is wild type action potential from a wild type mice versus an action potential from a mutated mice, and you see that the action potential, various different characteristics of the action potential are changing in the inhibitory neurons. So the firing in inhibitory neurons is selectively impaired, whereas the firing in excitatory neurons remains unchanged from the wild type to the mutated uh, mouse. And so we basically see that this specific gene mutation, which changes lysine to a threonine in humans, basically leads to probably in humans also leads to a impairment of inhibitor neurons, selective impairment of inhibitor neurons, whereas excitatory neurons remain unchanged, and because that is what they found in the mice, and probably this can help them further get at what might be possible therapeutic um, drugs or other interventions that can help prevent this from happening. Right. So the other important thing that we talked about in the class is the ethics of animal use in research. So transgenic animal research has advanced our understanding of several human diseases, but it is bound by very strict laws and annual reviews carried out by animal rights and ethics committee because most of these animals are put through a lot of stress and a lot of manipulative and often debilitating experiments are carried out on these organisms. So the major question here is that do we have the right to manipulate the genome of animals for research purposes? Because they are also again living organisms which might basically be living, feeling and have sort of understanding and basically behavior and so on and so forth. So how does our actions affect them? Basically. Right? So these are the questions that we need to be thinking about. We need to keep in mind when we are basically doing research um, transgenic animals. Okay. Does anyone have any questions till now? Great. So we'll move on to the practice assignment 5 for this week. So the first question is true breeding schemes are dash A organisms in which progeny are capable of breeding, B organisms in which parents are fertile, C organisms in which progeny are identical to their parents, D organisms in which parent and progeny are bred together. Can anyone answer? Feel free to type your answer in the chat or unmute yourself. Okay. So the answer here will be C, which is organisms in which progeny are identical to their parents. They are called as true breeding strains. Next question is in selfing of plants, we dash. So A, pollinate a plant with its own pollen. B, produce a plant that looks like itself. C, pollinate a plant with pollen from another plant. D, produce a clone from vegetative propagation. So the answer here is A, because in selfing, we are basically pollinating a plant with its own pollen. In crossing, we are pollinating a plant with pollen from another plant. The third question is repeating the same experiment as Mendel for a monohybrid cross. Scientists used 10 pea plants to study the trait tall. They observed that in the F2 generation, plants have a 2:1 ratio of tallest to short plants. The reason for this is because A, Mendel's experiments were done on strains of different pea plants. B, Mendel's experiments were done in Europe and recent experiments were done in India. C, 10 plants aren't enough to give correct results for a probabilistic process. And D, the 2:1 ratio is the correct explanation for the law of segregation. Okay, so the answer here will be C. 10 plants aren't enough to give correct results for a probabilistic process. Since Mendel carried out his experiment with a large number of pea plants, he was able to get at the ratio of 3:1, which we also mathematically see when we are using a Punnett square to get at the value. Here, since they are only using 10 pea plants, it's very difficult for them to get at the correct ratio, which is 3:1. Question 4, Thomas and Morgan Organ figured out that the eye color trait lies on the sex chromosome in fruit flies. True or false? The answer is true, as we discussed in class before. 5. If a set of parents have three offspring, why aren't the offspring identical? A. Because of recombination, gametes are different in both parents. B. Because parents have different genes. C. Because parents don't contribute at the same level for each offspring. D. Because the offspring reject different parts of the parent's genome. So the answer here is A. Because of recombination, gametes are different in both parents. We saw that during the process of meiosis, there's a recombination which produces four different kinds of gametes from each parent. Number 6, in forward genetics, we dash A go from genotype to phenotype, B go from phenotype to genotype, C go from wild type to mutant phenotype, D go from mutant to wild type phenotype. So in forward genetics, we are going from phenotype to genotype. So the answer here is B. In reverse genetics, we dash go from genotype to phenotype, phenotype to genotype, wild type to mutant phenotype, mutant to wild type phenotype. The answer here will be A, because in reverse genetics, they are going from genotype to phenotype. The screen for abnormal embryonic mutants in Drosophila melanogaster fruit fly is an example of dash. A, reverse genetics, B, Xantech cross, C, forward genetics, D, biotechnology. So this is the first example that we had discussed in the class. So the answer here will be forward genetics, because what they did is they basically got abnormal embryonic mutants and then tried to figure out which genes are causing these mutations. In site-directed mutagenesis, a mutation causes a change at one site randomly. A mutation causes a change in one base due to specific primer being used for amplification. A mutation is caused by substituting a T with a U. A mutation is caused by using non-standard bases. So the answer here will be E, because here in site-directed mutagenesis, a mutation is uh, causes a change in one base due to a specific primer being used for amplification. Number 10, in the research work of amplifier and Craig-Mello, RNAi can knock down gene expression by dash. A, not allowing gene transcription. B, blocking DNA replication. C, interfering with mRNA to block translation. D, blocking ribosomal activity. So the answer here is C, because amplifier and Craig-Mello discovered RNAi, which is basically a way in which we can interfere with mRNA and block the translation. Okay. Does anyone have any questions or doubts? If not, we'll stop here. Thank you for attending. I'll see you guys in next class. Bye.